on. I'm recording, so I'm going to hit it really quickly. And just listening to this, I think, is good for your learning and memory as well as reading it. So let me hit my first whiteboard here and just do really quickly. I'm going to do general types of muscle tissue and their characteristics. So this whole chapter is about muscles and muscle tissue. I'm going to start off with muscle tissue. There's three main types. We touched on this when we talked about the tissues: skeletal muscle, cardiac muscle, and smooth muscle. After I go over some general characteristics and some introduction, very little is going to be covered other than general characteristics of cardiac muscle. You'll learn all about cardiac muscle when you study the heart in A&P2. Smooth muscle we will do at the end of this chapter, so most likely I won't say anything today about smooth muscle other than a few general characteristics. Almost all of this chapter in almost all A&P books is about skeletal muscle. So let me just say a few things. Skeletal muscle is what you and I think of when we hear the word muscle. It's attached to bones, that's its location. It's voluntarily controlled. That means you have conscious control over its contractions. The tissue is striated. That means that it has stripes when we look at it under a microscope. And I'll tell you how they get those stripes later. But in one sentence, it's just from the banding patterns of their myofilaments, their proteins. And last but not least, the cells in skeletal muscle are multinucleate. They have more than one nucleus, and I'll also address how that happens later. Cardiac muscle only found in the heart. It's also striated, like skeletal muscle has stripes due to the banding patterns of the contractile proteins, but it is involuntarily controlled. So we don't have conscious control over our heartbeat. We can't just sit here and think, oh, I want to speed up my heart contraction, and it doesn't. It doesn't work that way. Smooth muscle, it gets its name because there are no striations in the cells. There are no banding patterns or stripes. Smooth muscle is found lining the walls of organ, mostly digestive organs, but respiratory organs as well, predominantly lining organ walls and vessel walls. So in our arteries and veins and many of our internal organs, smooth muscle lines the walls and allows for contraction, for movement of things. So those are the three main types, and they're very brief general characteristics. Now I'm going to this is almost the same in all A&P books. And I'm just going to list some major functions of muscle tissue in general. It's not any one of these three in particular. The first one is always movement. That's the biggest, one of the largest and most unique characteristics of muscle tissue, is that it produces some sort of movement. Either externally, I move my arms and legs, or my mouth like I'm talking now, or internally as well. Muscle also maintains posture and stability. So I can sit up straight right now, and you can also, I assume you're probably sitting, can also sit up straight. You don't even have to really think about this because of muscle contractions that hold your head up, keep your spine erect, yada, yada, yada. A third function is sort of the same as this, but it's joint stability. So I'm going to write joint stabilization. I sometimes list those as the same thing. This mostly refers to, though, not necessarily maintaining a posture, but just those synovial diarthrotic joints that we went over last chapter. They become much stronger with muscles that insert or originate in those joint areas. And finally, last but not least, muscle contractions produce heat, body heat. And that's predominantly skeletal muscle, but all muscle produces some contraction, produces some form of heat, mostly skeletal muscles. You go outside on a cold day, like yesterday, t-shirt and shorts, you start shivering. Those are muscle contractions that raise body temperature. brief synopsis of general characteristics of muscle tissue. Almost all muscle tissues have the characteristics of excitability. And what that word means in A&P context is the muscle cells themselves have the ability to generate an electrical impulse, some sort of electrical activity. So we need the nervous system, or in the case of smooth muscle and cardiac muscle, as well as the endocrine system, to produce muscle contractions. But the muscle tissue, the cells themselves, also produce some sort of an electrical event. They're excitable. That's where that word comes from. These cells also have contractility. That just means, like the word says, the ability to contract or shorten. And that usually produces tension, gives the ability to move something. There's also something called elasticity, which means their ability to recoil once they are stretched. They're also, I'm just going to write stretchability. They can stretch and recoil. All right, so let's move on now. Pretty much from this moment forward, everything that comes out of my mouth for the rest of this lecture is going to be about skeletal muscle tissue. All right, so from this moment forward, if I don't say skeletal muscle, if I just say muscle, I'm referring to skeletal muscle, this stuff that you and I think of as muscle that's attached to our bones that we have voluntary control over. So let's move on to a new whiteboard. And this is how a skeletal muscle is arranged. So I'm going to write muscle arrangement. A muscle is arranged, a tube wrapped with another bunch of tubes, wrapped with another bunch of tubes. It's, I always give this analogy in my lectures. It's like when you walk into Lowe's or Home Depot and you see all these pipes bundled together. That's really what a muscle looks like structurally, anatomically. So individual muscle cells are long and cylindrical like this. So it's a bit of a unique characteristic that they're so long and cylindrical. And we call an individual cell a muscle fiber. So if you read or hear me say the word fiber, I'm just referring to one individual skeletal muscle cell, a muscle fiber. Fibers are long and cylindrical like this. And the macro arrangement, that means without using a microscope, what we can see with our naked eye is each individual cell is wrapped in a thin, delicate connective tissue covering called endomysium. That's thin, it's reticular fibers mostly. I'm just writing thin, delicate CT. It wraps each individual fiber. So if I just quickly put a wrapping, let's make it yellow. If I wrap this in a thin, delicate coating of endomysium, there it is. Then these individually wrapped cells get bundled together and wrapped again as a bundle in a thicker connective tissue covering called perimysium. So this is really dense, irregular CT. So I'm just going to write dense, irregular CT. It's thicker and coarser than endomysium. And what this stuff does is it bundles together a whole bunch of individual cells. So I'm going to quickly make a bundle of individual cells. Each one of those individuals has already been wrapped in endomysium. Now we wrap that bundle in a thicker connective tissue covering like this. and we call that perimysium. All right. That bundle that was created by the wrapping of the perimysium is called a fascicle. Fascicle. Just think of, I'm writing the word bundle. So the wrapping of perimysium actually creates a fascicle, which is just a bundle of individual muscle fibers that have been bundled together. Finally, what makes up the entire muscle is a thick, coarse connective tissue covering called epimysium. This is also dense, irregular CT, but it's thicker and coarser than perimysium typically. And it wraps the entire muscle. So what it does is we wrap several fascicles together to form a muscle. So you can see that a muscle that you can point to and touch to on your body is really individual cells wrapped and then bundled together, and then several bundles, several fascicles bundled together a last time to make a muscle. So I'm going to call up a new whiteboard here and just quickly, so here are individual fibers. They've been wrapped in our perimysium. Now I'm going to make another bundle of individual fibers. It also is wrapped as a bundle in perimysium. And then if we wrap these together in a really coarse connective tissue covering called epimysium, and this, especially the epimysium, this connective tissue runs off the end, but it bundles several fascicles together to form a muscle. Which brings me to the next term that I'd like to run across for you, and that's tendon. The definition of a tendon is really just a rope-like or rope-shaped connective tissue that connects a muscle to a bone. That's sort of definition of tendon. How it's created, how a tendon is created, is these excess wrappings. And technically, it's all three of the wrappings that we discussed on the last page, but predominantly, it's epimysium as it comes off here on the end. This would be a tendon, and it attaches, that's what attaches to a bone. There's the head of the femur. This word, aponeuroses, it's a funny word. Aponeurosis is singular, ES would be plural. This is, just think of a flat sheet-like tendon. That's what it is. So flat muscles don't have a rope-like structure like this, circular or tube-like. Flat muscles, their connective tissue coverings come off in a flat manner. So I'm just going to write flat sheet-like tendon. And they're associated with flat muscles like our abdominal muscles or the frontalis muscle on the forehead. And quickly, I'm going to identify nerve and blood supply. So I'm going to use my muscle drawing up above. Typically, not 100% of the time, but typically there is a major nerve that comes in to stimulate this muscle, as well as a major artery that comes in to supply blood to this muscle. And there may be one or two sort of major veins that drain blood away from this muscle, like this. So let me change this arrow so you don't get confused. Now, as far as the innervation goes, the nerve, all skeletal muscles typically have a major nerve coming in, but that nerve quickly, as soon as it hits the muscle surface, it branches hundreds, if not thousands of times. And a branch of this nerve goes to each and every individual cell. Skeletal muscle cells must have their own nerve ending in order to contract. That is imperative. They won't contract without that direct neural stimulation. That's not really true for cardiac muscle and smooth muscle. Likewise, this major nutrient artery that comes in, just sort of like same with the diaphysis area of a bone. It also branches very quickly and serves vascularization all over through it. So these skeletal muscles are highly vascularized as well as heavily innervated, even though from the outside it may look like just one vessel going in and one nerve going in. That's really misleading visually. Now, your book does briefly talk about attachments. I was going to talk about that later, but I'm going to say them now. Attachments just means the tendon ending that attaches to bone here, and on my make-believe muscle up here, it also attaches just, let's say, to another bone here. Those are points of attachment, and we call those points of attachment a muscle's origin and insertion. The difference between these two terms refers to what happens at that point of attachment when the muscle contracts. In general, I can't think of an exception off the top of my head. In general, the origin of a muscle is the end of attachment where when the muscle contracts, there is no movement or very little movement. The insertion end is the end of attachment where there is movement when the muscle contracts. If not all of the movement, then the vast majority of it. So take, for example, your biceps brachii in your upper arm. It's attached up behind your shoulder to the scapula. That would be its origin, and its insertion is down in your forearm on the radius. When that muscle contracts, your forearm moves. So it's the point that's attached to the radius that's the insertion, and the origin is up on the scapula where there is no movement when that muscle contracts. All right, now we need to look. We've looked at the macroanatomy of a muscle. I'm going to quickly do the microanatomy of a muscle cell. So I'm going to draw a really big muscle fiber cell, and we're going to talk about the anatomy and a little bit about the physiology of what's in this fiber. So remember, an individual cell is just called a fiber. We talked about how a muscle is arranged. Now let's look inside of the fiber. Ironically, or I should say, almost coincidentally, it's a better term, what's inside the cell looks like what's outside of the cell, which can be confusing. So if we dissect this individual fiber, what we find inside, guess what, are a bunch of bundled tube-like structures. There are typical organelles in here. There are nuclei, there are mitochondria, ribosomes, all of those things. But what takes up most of the space inside are these specialized organelles called myofibrils. The prefixes myo and sarco mean muscle. So we have, this just means technically really small muscle fibers. They're not, these are actually, I'm going to write this out. They're specialized organelles. And they're found within an individual fiber that contain contractile filaments. What a contractile filament is just a long stringy protein that actually produces the shortening, the contraction of the muscle. And to make things even more confusing, I'm going to blow up my myofibrils here now. Here are my myofibrils coming out of that muscle fiber cell. Within those myofibrils, these are the myofilaments. They look like little threads. Now you can see it just looks like tiny rods bundled together and bigger rods bundled together in a bigger rod. These threads that I've, contractile filaments are also called myofilaments. So, and it's these tiny threads that I'm drawing right now that actually do the contracting process. A few more things about this cell. we call the plasma membrane the sarcolemma. Sarco means muscle, lemma just refers to plasma membrane. So that endomysium that I discussed back here, the yellow stuff around each individual muscle cell, is actually on top of the sarcolemma. So that endomysium would be above or on the outside of the plasma membrane like this. We call the cytoplasm, the fluid-filled cytoplasm inside of the muscle cell, the sarcoplasm, which just means, let me get black again. So if you read or hear me say the word sarcoplasm, that just refers to the inside of the cell, the cytoplasm of a muscle cell. Oftentimes what we find in here, other than it's stuffed full with these myofibrils, like I said, there are nuclei. They actually get pushed out to the edge. There's so much stuff in there. But there's mitochondria and a few specialized things. One of those specialized things, glycosomes, are these little granular, they're storage containers. They're considered an organelle, but glycosomes, they store glycogen, which is just sugar, so that the muscle cell can produce ATP. It's a source. Also in the sarcoplasm, there's something very specialized called myoglobin. Remember, we're inside of an individual fiber or cell here. Myoglobin sounds a lot like hemoglobin because it behaves a lot like hemoglobin. This is a protein that binds oxygen, just like hemoglobin in the blood. And really what it does is it binds oxygen, it gets it from that blood supply, and it stores it. It stores oxygen for use when the muscle contracts in an aerobic fashion. I'm going to skip now the banding patterns of a typical skeletal muscle fiber because I think it's more critical that you understand how a muscle contracts prior to understanding that banding pattern. So this is going to take pretty much the rest of the lecture, but I'm going to tell a really, really long but interesting, I'm saying that in quotes, story about how a muscle contracts. And it's going to take me a couple whiteboard pages. It's going to take your patience to listen to this. So let me call up a whole new set of boards here. I'll do that first. And I'm going to start at the very beginning of how a muscle cell contracts, and then we'll keep going till the very bitter end. I'm drawing my muscle fiber cell, and I put a little indented space on the surface for a reason. I'm going to make my muscle cell a little wider so it gives me more space to draw inside. This is an individual muscle cell, and it would run off the page because it's so long. Now I'm going to draw the end of a neuron, a nerve cell. That's the end of the nerve cell that's actually innervating or exciting this muscle cell. First thing you notice, there's no physical connection. That's true. There's a fluid-filled gap there. Let me label this neuron. So this Bart Simpson's foot-looking thing is just the end of that nerve cell, the neuron. And there's many of them going to different cells. We'll talk about the nervous system at the end of the semester, so I'm not going to get into how the nerve cell generates an electrical impulse, but it does, and it sends an electrical impulse. That electrical impulse is called an action potential. Just think electrical stimulus. So I'm going to draw that. Here comes an electrical stimulus down the edge of this nerve cell, and I'll label that an action potential. That just means electrical stimulus. When that electrical stimulus gets to the end here of this terminal, it's called the terminal end or terminal button, that electricity causes gaps to open up on the membrane here. So I'm drawing little doors opening up. These little pores or gaps or channels open up and allow calcium ions to flow in. I'll review all of this at the end. So don't feel like you have to grasp this the first time. I would watch this multiple times. Now, when this nerve cell is not firing an electrical impulse, what it's doing is it's manufacturing and storing a chemical. And I just made a bubble with little dots in it. That represents a vesicle with a chemical in it called neurotransmitter. Neurotransmitter just means a chemical that a nerve cell releases. So there it is. The chemical is inside of this storage vesicle. When calcium comes in to this neuron, it causes this vesicle to migrate to the end and release its chemical into this space. The chemical that is released 100% of the time in all skeletal muscles is called acetylcholine. ACETYL. That is the specific name of a neurotransmitter. It's used in other places in the human body, and always in a motor neuron innervating a skeletal muscle cell. That's just the specific name of the chemical that was released. There it is, here it is, diffusing into this space. By the way, this space is called the synaptic cleft. Cleft just means space. Synapse refers to this whole junction between the nerve cell and the muscle cell. So we call this space, this fluid-filled space, the synaptic cleft. As acetylcholine diffuses across this space, it then binds to receptors on this indented portion called the motor end plate. So I'm going to label these receptors. They're specifically acetylcholine receptors. Receptors on motor end plate. None of this stuff is super complicated. It's just a story that has a lot of new names. That's it. So most of this is language. I'll summarize at the end. When the acetylcholine chemical binds to receptors, I'll exaggerate it. When it binds to receptors on the motor end plate, something magical happens. It causes this muscle fiber to generate its own electrical impulse on the surface, like this. So we've gone from electrical to chemical to, and when that chemical binds, it causes electrical to happen again. So on the sarcolemma, which means plasma membrane of this muscle fiber, we have an electrical impulse generating outward from this point. There are organelles called T-tubules, and you'll see why they're called T-tubules, because they look like the letter T, and they're tubes. The T actually stands for transverse tubules. These T-tubules are simply, what they really are invaginations in the sarcolemma, the plasma membrane of this muscle cell. Their sole job, these T-tubules, their sole function is to conduct this electrical impulse from the surface of the muscle cell inward to the inside, like this. So the electrical impulse, which is called an action potential, travels down these T-tubules to the inside of the muscle cell. Now, when it gets to the end of the T-tubule, there are two storage sacs associated with each T-tubule, and they are just containers full of calcium ions. Remember when we studied the bones, we said calcium is the most used mineral, ionic mineral in the human body. This is why. You can see we used it here. Calcium is also stored inside of these red storage containers. Those have a fancy name, as like everything else, but it's not complicated. They are just storage containers. So I'm going to write the name out in red. They're called the terminal cisternae. It's just Latin. Terminal cisternae of the sarcoplasmic reticulum. Terminal cisternae just refers to cisterns, these big storage cavities at the terminal end of the T tubules. So this terminal cisternae of this network of tubes called the sarcoplasmic reticulum. Sorry, I'm writing with a plastic pen on a glass board. The sarcoplasmic reticulum network just means there's a tubular network that connects these things together. Really, they're continuous, they're not individual. The bulging sacs are the terminal cisternae. They store calcium ions. So I'm going to repeat this story really quickly. Electrical impulse travels, let me get my black pen back. Electrical impulse travels down the nerve cell. When it gets to the end, it causes calcium ions to influx into the cell. That causes this neurotransmitter acetylcholine that's stored in a vesicle here to be released into the synaptic cleft. The acetylcholine diffuses across the synaptic cleft and binds to motor receptors on the motor end plate here. The binding of acetylcholine actually causes a new action potential electrical impulse to be generated along the surface of the muscle fiber. That electrical impulse needs to get inside of the cell, and it's T tubules that guide that electrical impulse inward. And when the electrical impulse reaches this area, it's called the triad. It's only called a triad because there's 2 terminal cisternae associated with each T tubule, so 2 + 1 is 3. When the electrical impulse reaches this triad region, it causes calcium ions to be released inside of the cell from those storage containers. Calcium floods the inside of a muscle cell. So when the beginning of the story is when a nerve cell stimulates a muscle cell, a skeletal muscle cell, calcium is eventually released in huge quantities inside of the cell. So I'm going to save that board, and then we're going to find out what happens to this calcium, okay? So let's hit a new board. Deep, deep inside of the muscle cell, under a gazillion power of magnification, there are little myofilaments, they're called. They're just stringy proteins that sort of resemble this. One of them looks sort of like a golf club that's attached to a center structural protein element. I'm going to simplify. I'm just going to draw 2 of these. It's a simple version of what's really going on. We call this structure here, that's called the thick contractile myofilament, the thick filament, which we refer to as myosin. That's the thick filament. Also arranged is, I'll use yellow. I've exaggerated its size so that you can see it, but it's much smaller. This is called the thin filament, actin. Actin proteins function in more than just in muscle contractions, but this is what we're worried about right now. So myosin's the thick filament, actin is the thin filament. To cut to the end of this story, myosin is going to reach up, grab actin, and pull it this direction toward this center line here. We call this the M line. It's just M just stands for middle. And this would just be an opposing myosin. I'll draw it really quickly. There's also, here's an actin associated with it. It's much more complicated than my drawing, but I think a simpler diagram will help you understand this process better. So the end of this story is myosin reaches up. Let me use black. Myosin is going to reach up, grab actin, and pull it this way. Likewise, this myosin is going to reach up, grab this actin molecule, and pull it this way. When we pull thin filaments in this direction over and over and over again, if I have this setup, and then I have it again here, and then I have it again here, and then I have it again here, thousands of times throughout along this muscle fiber cell, and we're going to pull this actin molecule this way, and do it again here, and do it again here. I'm traveling down a muscle cell now. That shortens the entire cell. What it does is it makes the cell go from this shape to this shape. That's a muscle contraction. It's a shortening of the entire tissue. So that's the goal, is for myosin to reach up, grab actin, and pull it toward the M line, like this. Now we're going to really look at how it's able to do that. So I'm going to draw a new actin molecule. Actin, the reason I'm drawing it like this is it's a bunch of these just small it's an amino acid chain. It looks like a bead of pearls almost. And on each of these individual amino acid sites or peptide sites, there's an active binding site. So how that myosin, that red golf club, can reach up and pull on these, pull the actin, is it binds to a specific site on actin. So what I'm drawing here are binding sites. They're called active sites. When our muscles are relaxed, so let me draw my myosin molecule. There it is. It wants to reach up, attach to a binding site, and then pull the actin molecule this way. When our muscles are at rest, these binding sites are covered up by a different protein. And it looks like a grapevine just sort of wrapping around the actin molecule. This protein that I've drawn, the grapevine protein, is called tropomyosin. Its sole function is to cover the active sites, the active binding sites on actin. So when our muscles are relaxed, it looks like my picture right now. So we need to uncover those binding sites in order for myosin to reach up and grab it like this. On the surface of this grapevine are these little tabs, these little short proteins like this. Okay. These little tiny proteins that are attached to tropomyosin are, this is confusing, I get it, they're called troponin. Calcium, this is really important, so I'm going to say this really slowly. Calcium ions bind to troponin. So remember back here, in this diagram, the whole goal to the first part of our story was to release these calcium ions inside of the muscle cell. What happens with those calcium ions, those calcium molecules, is that they bind to troponin, and when that happens, they lift the whole grapevine off of actin and expose the binding sites so that myosin. So what generates at the very beginning of this story of muscle contraction, we have nerve, electrical impulse, chemical release, electrical impulse on the muscle cell. Eventually we get calcium flooding the inside of the muscle cell, and it's these calcium ions that then bind to these little green tabs here. They're not really green, I just drew them green. And that binding process lifts the blue grapevine off of the actin molecule so that our binding sites are now exposed. I left one exposed right here so that I could, the myosin molecule, it's called the myosin head, like the head of a golf club. It reaches up and can bind now to the actin molecule and pull it this direction. And like my previous picture here, this myosin reaches up and binds, this myosin head reaches up and binds when that calcium exposes the active site. And then the actin molecule, the yellow one, gets pulled toward the M line this way. Now, let me go back to my most recent picture. This thing, the myosin heads, needs energy in order to do the pulling action. We're almost finished with this story. In order for this head to, it reaches up and binds to actin, but in order for this head to actually pull, have the energy to pull, it gets that energy from an ATP molecule, which is the next discussion. Your book calls it forming a crossbridge, the crossbridge cycle. So I'm going to go through that in detail. When a myosin molecule is relaxed, it sort of looks like this. Okay, I'm just going to draw the actin molecule up here. Let's just assume that the nerve cell stimulated the muscle cell. Calcium was released. The active sites on actin are exposed. Now, myosin will reach up, grab actin, but where it gets the energy to pull is from this molecule right here, which we talked about the first week of the semester. Adenosine triphosphate, or ATP, means it has 3 phosphates attached to it. That's the energy storing molecule that energizes most things in the human body, including this process. And here's how it works. An ATP molecule that the cell has made, the muscle cell has made, that attaches to the myosin head. There it is. Then this ATP molecule that's attached to the myosin head splits its terminal phosphate. How that happens is there's an enzyme called ATPase, which is an enzyme that hydrolyzes. That just means break something apart using water. It hydrolyzes the ATP molecule, and we break the last phosphate off of it, and we end up with ADP plus P. That just means we peeled one of the 3 phosphates off of ATP, and now we have 2 phosphates and 1 phosphate. So I'm going to draw an arrow here. When that splitting happens, the head of the myosin molecule goes from this position to this position. So we've moved it this direction. And the ADP and P are still attached to the head. The energy, it's like cocking a gun. I always tell students, if you pull this head back this way, like cocking a gun, the little thing on a gun, I don't even know what it's called, you have given that gun energy to do something. Likewise, when this ATP molecule splits into ADP plus P, so I'm going to draw them still attached here, ADP plus P, it makes the head go back like this and gives it energy. Now, when that head, the head that's cocked like a gun, it looks like this. Now, when it reaches up and grabs this molecule, it has the energy to pull forward this way, and the actin molecule gets pulled. So that happens. This energized ATP molecule reaches up, attaches to the active site on actin, pulls the actin molecule toward the M line, and we have a situation that kind of looks like this now. It's already been pulled. So we've gone from here to here. The ADP and P then fall off. So they leave that myosin head, ADP plus P. But this connection remains. And it doesn't unlock, this binding doesn't unlock until a new ATP molecule attaches to it. And when a new ATP molecule attaches to it, this bound myosin head lets go, and the whole process starts all over again. So it lets go, it has a new ATP molecule on it, then it lets go of the actin, and then that ATP splits from this enzyme. The head cocks again, it reaches up and pulls the actin molecule even further. It keeps repeating this process like the ratcheting of a wrench almost. So it pulls a little bit, lets go, gets a new ATP molecule, lets go, cocks again, reaches up, grabs, pulls it a little bit further. ADP and P are released, new ATP attaches, it lets go. That new ATP splits, it reaches up again, grabs, and keeps pulling it in little tiny increments toward that M line. And then in the opposite direction, we have actin molecules coming this way. And just like I'm going to hold my fingers up here, if I take my fingers like this and I pull and I push them together like those actin molecules are going like this, that shortens the entire cell. And that's what a contraction really is. So before we stop this great and long story, I'm going to say a few more things about this relationship. We call this whole, the end of this story, when the actin molecules get pulled toward the M line. I want to go over this language with you because I will use it and your book uses it. We call it the sliding filament model of contraction. Okay. This term, this long term, just refers to the process I just described of myosin reaching up and pulling actin. And what happens is those actin molecules visually appear like they're sliding past one another, crossing over one another, moving toward that M line and shortening the muscle. So a little bit more of the language. Remember one of my first diagrams of the nerve cell and the muscle cell. I'm not going to go reiterate this whole thing again. You know that acetylcholine is released into this space and it binds to receptors down here. We don't typically want a muscle to stay contracted. If this neurotransmitter is not cleaned up right away, excess neurotransmitter, the muscle will want to stay contracted. And that's usually not a good thing. There is an enzyme that hangs out in this synaptic cleft area. And I always draw the enzyme. I know this is cheesy. I always draw the enzyme like a little Pac-Man because that enzyme comes in almost as soon as this neurotransmitter is released. This enzyme comes in and it degrades, it gobbles up excess acetylcholine, that neurotransmitter. That allows the muscle contraction to stop. It's one factor that allows that muscle contraction to stop. This enzyme that cleans up that excess neurotransmitter in that space is called acetylcholine esterase. So it's two words mushed together. Acetylcholine esterase is the enzyme that degrades excess acetylcholine Acetylcholine, by the way, is abbreviated AC little h. So there is the bulk of the story that you need to learn for the first half of this book. The last thing I'm going to say before I cut this lecture off is my drawing of the myosin and actin molecules is somewhat simplistic. So I'm going to quickly draw a new picture. I'm going to use red. So if we use red to draw this myosin, here's the M line. I'm going to give you a little bit better idea because your book drawings are more sophisticated. I'm going to complicate my drawing a little bit, but now you know what everything stands for. Here are the horizontal thick myosin filaments. And they don't just have a single golf club head at the end. There's multiples of them because there are multiple myosin molecules. This is what I meant by more complicated. So if you had multiple golf clubs stuck together, the shafts of those golf clubs stuck together, their heads would kind of flare out like this. And then we have several actin molecules associated with those myosins. So I'm going to draw a pattern. This is a structural protein that holds those actin molecules horizontally in orientation with myosin. I think I used yellow for my actin molecules, so I'll stay consistent and do that. These are the actin molecules that are associated with the myosin molecules. And what I've drawn here is called one contractile unit, called a sarcomere. This word sarcomere refers to one contractile unit of myofilament arrangements. The reason is because there would be new actin molecules here. That this blue zigzag, by the way, has a name. It's called the Z disc. It's just a structural protein that holds those actin molecules together. So one sarcomere actually goes from one Z disc to another. Nevertheless, there are new actin molecules to the side here, and this pattern just keeps repeating. We would have another M line here with myosin molecules. Doing this quickly. And it would repeat again over here. It repeats thousands of times throughout the individual cell. Here's a new M line. Here's a new myosin molecule. And it just keeps repeating over and over and over again. You'd see the actin molecules coming in here. From Z disc to Z disc is what we call a sarcomere, one contractile unit. Now you can see this regular arrangement of myofilaments. This pattern that goes here, 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 and the overlapping of these filaments in this area is what gives the muscle cell its striated pattern. So when you look at a muscle cell under a skeletal muscle cell under a microscope, it has this very regular banding pattern like this. Where that visually comes from is the overlapping pattern of these sarcomeres and these myofilaments within those sarcomeres. There are names to the different parts of the sarcomere that I try to tell my students not to get totally hung up on. There's things called the H zone, the A band, the I band. I think it's much more important that you understand that from here to here is a sarcomere, and that it's this overlapping, consistent pattern that gives a skeletal muscle its striated pattern or striated look. So I'm going to cut it there for this mini lecture, and let me cut my recording.